The streetlight effect goes as follows. You lose your keys in the dark, and the only place you go to look for them is under the street lamp because that's the only place that you can see. Now, you don't know where you lost your keys, but you only look under the street lamp because it's dark everywhere else. Well, in the field of metabolomics, for example, one could identify only a small fraction of the total available metabolites. So what do? You do a targeted analysis. You look for certain metabolites. You look for the ones that you can see, but there's this whole 90% of them, perhaps, that you can't see. And so by using this technology where we can identify the large majority of metabolites, for example, and we believe that we can, it opens up a whole new world of investigation because you can look anywhere for your solution and not only under the street lamp.
Welcome to the Smart Biotech Scientist. I'm David Brühlmann, and this is part 2 of our conversation with Tom Rizzo, who is a professor emeritus at EPFL and now CSO at Isospec Analytics. Last time, we covered the science behind cryogenic infrared ion spectroscopy, and today we are getting practical. How this technology helps you discover biomarkers, identify mysterious degradation products, and accelerate process development. We'll also explore Tom's transition from academia to entrepreneurship and what it takes to commercialize breakthrough technology. If you're dealing with complex mixtures and unknown impurities, stay tuned. This is for you. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. This new dimension, what will it enable scientists, companies, pharmaceutical companies, or also, I'd say, for even biomarkers or so, what will it enable? Can we detect new molecules we were not able to detect before, or will the workflow be faster? Or I've done a lot of omics also in my career, which can be very complex. I imagine your technology could simplify that too.
Okay, well, there's several parts, I think, to my answer in this. One is, in the area of glycans, for example, there's a real difficulty in analyzing different isomers. And we know, and there's evidence in the scientific literature that, for example, if you have the glycosylation of a monoclonal antibody affects its efficacy and safety and its lifetime. But if you can't resolve the different isomers, you don't know what's responsible for that difference in efficacy. And so being able to do isomer-specific and glycan analysis sheds new light on the mechanisms for, or the effect of glycosylation on the efficacy of these kinds of things. So that's one side of things. That is, being able to determine to the isomeric level what species are adorning your protein is an important part of understanding its function and why it functions so well. But there's a broader issue, particularly when you talk about different omics, metabolomics, for example. And I like to use the analogy of the streetlight effect. Do you know what the streetlight effect is? Have you heard of this before?
No, I don't know.
So, okay, the streetlight effect goes as follows. You lose your keys in the dark, right? And the only place you go to look for them is under the street lamp because that's the only place that you can see. Now, you don't know where you lost your keys, but you only look under the street lamp because it's dark everywhere else. Well, in the field of metabolomics, for example, one could identify only a small fraction of the total available metabolites. So what do? You do a targeted analysis, right? You look for certain metabolites. You look for the ones that you can see, but there's this whole 90% of them, perhaps, that you can't see. And so by using this technology where we can identify the large majority of metabolites, for example, and we believe that we can, it opens up a whole new world of investigation, right? Because you can look anywhere for your solution and not only under the street lamp. So I think that's an apt analogy to the situation, because current techniques can only identify such a small fraction of biological molecules, metabolites in particular.
Yeah, and I think it will open a lot of avenues in disease detection, because I think you're making such a good point. And if you go to the doctor, they have their program, and we pretty much have measured the same biomarkers for many years. But now I think with expanded capability, we'll be able to pick up a lot more metabolites. And I think now on top with AI, I imagine the possibilities will just be endless. So I just have a follow-up question about where a bit you think AI will take this. And also, I think another very practical aspect is that with omics or with mass specs, you need huge libraries usually to then correctly identify your molecule. How does that work with your technology?
You're right. If you want to identify a molecule from its infrared spectrum, you need to have a library of infrared spectra. And in fact, we have mechanisms, normally when you do that, you would think that you need a standard. And getting standards for different isomeric molecules can be extremely difficult, or if not impossible. So this could potentially be a problem, but we've developed techniques to interpret infrared spectra of molecules without having a standard. And how it works is, let me take the example of glycans, because that's one that we've done a lot of work on. You measure the infrared spectrum of a parent molecule, and you find that it's not in your database of spectra. Well, what do? What we do then is we fragment the molecule. We can measure the infrared spectra of the different fragments. Usually you can find fragments that are characteristic to one isomer or another. So if you're looking for a substitution on one branch or another branch, and you break it off, and then you see just the branch and not the rest of the molecule, and you see that species is on there. And for that smaller molecule, we say the infrared spectrum, is that in our database? If it is, then you've identified that. And then you can say, okay, we've identified now the parent because we know that substitution was on this branch of the molecule. If after the first fragmentation, you still can't identify the molecule, you can fragment it again, measure the infrared spectrum of that molecule, and you can do that until you have small enough species that are in our database. And then we work our way back up. Once then you've identified the infrared spectra of the parent molecule, you don't have to go through this fragmentation process again. You just do it once. You've now identified it, it's in your database, and now if you have a still larger molecule which fragment back to that one, then you only have to go to the point where you found the fragment in your database. And so we have a mechanism by which we grow the database from smaller species to larger species. And we do that by taking a large molecule, cutting it back until we get things for which we have in our database. So that's the general procedure of how to do that for a large, more complex molecules. Now, in the case of smaller molecules like metabolites, there we need to rely to some degree on imputed spectra, quantum chemistry calculation. But for smaller species, smaller metabolites, quantum chemical calculations are really quite good. And so that's what helps us on the smaller end of the spectrum. And there are now AI-based techniques that help quantum calculations, matching those with measured infrared spectra. And so that's one way that we will be using AI, is in combination with quantum chemistry calculations to match those with the measured infrared spectra. But we use AI in other ways as well, because this is another part of your question, is how we incorporate AI technique. Once we get all this information about metabolites, for example, what do we do with it? How do we use those measurements to increase our understanding of disease mechanism? And there we can use AI together with all the information with other more traditional omics techniques, for example, to put it all together and help medical researchers understand the mechanisms of disease. And so we really think AI will help us bridge the gap between analytical measurements and understanding disease mechanisms.
Yeah, that's where I see both the opportunity and the challenge, because I have had several conversations with my former boss about omics, and it was always the question, yeah, it's great. We are able to generate tons of data, but it's very difficult to make meaningful conclusions out of that. What does that actually mean? And for instance, at that time, we were optimizing bioprocesses. okay, we see that there are changes, but what does that mean? What do we need to change now to actually make it better? So I think AI will definitely help us make much better decisions. So my question now is, what is your vision of the technology? And also, as you're combining this with AI, with other technologies, where do you see this going in the next few years?
We see it going in a couple different directions. One is we're establishing a platform that we can provide, basically a service platform, to help medical researchers, pharmaceutical companies analyze biological samples with the hope of learning more about disease, understanding disease mechanisms. And so, on the one hand, we provide a service that people could send us samples and we could not only give them data, like give them an Excel sheet with what's in their stuff, but now using AI, help them or give them the tools by which they can start to analyze these disease mechanisms, if you will. We would provide then a platform, not just data, but a platform with AI tools as well to help analyze these things. On the other hand, we actually want to put these tools in the hands of researchers themselves. Like I mentioned before, we're working together with Agilent to produce a commercial instrument by which people can get their hands on this technology and apply it in ways that perhaps we can't even imagine. And so we see it going in that direction. And then we have this ability to also produce databases of infrared spectra using our current instruments. And so we could then provide a service of developing specialized databases in different domains because we have that expertise.
Before we continue, Biotech founders, I want to talk directly to you. Do you know that feeling when an investor asks about your manufacturing strategy and you freeze? One wrong CMC decision can cost you $2 million and 18 plus months of delays. Yet many CEOs are flying blind on these critical decisions. That's why I created the 1-week CMC strategy assessment. In 1 week, you get a personalized assessment of your molecule and development stage. We identify critical gaps before they become million-dollar mistakes, plus a strategic roadmap for high-stakes investor meetings. One CEO told me, David took me from feeling powerless about our CMC roadmap to confidently pitching investors. An absolute game changer for time-pressed CEOs. Whether you are preparing for Series A, approaching IND filing, or facing scale-up challenges, this gives you confidence to ace investor and board conversations instead of surviving them. Ready to stop second-guessing your CMC strategy? Check the link in the show notes and secure your spot. Because your breakthrough therapy deserves a solid CMC strategy. Now, let's get back to our episode. When do you think will this device be available? Are we talking months, years, or what is your current timeline?
So we currently have a service that we offer.
The service, yes.
And so we're working with hospitals. We have collaborations with hospitals here in Switzerland, in Germany, and in Belgium. But for people to get their hands on the instruments, it usually takes a couple of years to go from a prototype to a commercial instrument. Maybe available relatively quickly is an add-on to existing instruments. But for a fully integrated instrument, I would say we're still a couple years off.
Now, I would like to bridge now your academic career and now your, I'd say, entrepreneurial career. A lot of our listeners, they are either in academic or they startup founders. So I would love to have your take on this. You have seen now both worlds. You have seen also the transition and you've talked to a lot of scientists during your career at EPFL. So what advice would you give a scientist, a PhD or postdoc who would now like to start his or her company?
As I said before, it's a completely different world, right? And there's a lot to learn, and I'm still learning quite a bit. You have to really be passionate about what you're doing. It's not for the faint-hearted. Many startups just don't make it, and one has to go in realizing that. But from a very specific practical thing that we've experienced, you see, we only had 1 year overlap between my laboratory and the company because of my retirement. So once you're out on your own and you no longer have that academic laboratory, you don't have the ability to do R&D in the same way as you did before. And that's a real drawback. R&D in a startup is tough to do because investors want you to show how you can make money. And just doing R&D to improve your technique doesn't generate income. If a professor spins off a startup and that professor continues his laboratory and can do more fundamental R&D at the same time as the company is trying to commercialize it, that's a real advantage because the weight of doing R&D in a startup, it weighs pretty heavily on the company. And then there's competing interests. Someone wants to use the machine to understand a little better the technology, but you have to produce for your customers. So that's one practical piece of advice I can offer.
Excellent. And Tom, in the beginning of our conversation, you were talking about what is the legacy, what is the purpose of this, say, new season as an entrepreneur. So I'm really curious about that. You decided to start this adventure with the company. What is giving your sense of purpose now, and what legacy do you hope to build through your entrepreneurial chapter now?
As I look at things now, what drives me to some degree is my life experience. My father died of colon cancer. I've had friends with prostate cancer. My wife has had leukemia. And, as you go on your career and you get older, you see friends, people who suffer from these things, which, if diagnosed early enough, could have been treated in a way. Fortunately, my wife's leukemia was treated with the stem cell transplant, which was completely successful. And so she's 100% in remission, and we're by that. And so the whole idea of early diagnostics is something that kind of drives me. Can we find biomarkers that would allow us to determine years before a disease really develops? If our technology just helps the early diagnosis of one of these diseases, then I think it would be a great success. So that would be a great legacy to leave, I think.
Wow, I love that. I love that. Very powerful. This has been great, Tom. Before we wrap up, what burning question haven't I asked and that you're eager to share with our biotech community?
I think you've covered it pretty well, David. Offhand, I can't think of any burning question that you haven't asked. Actually, it's maybe not a burning question, but one question which you didn't, it's a technical question about the technique. You didn't ask me how we actually measured the infrared spectrum.
Oh, yes. Good point. Yes.
Now, if you think about it, how can you make a technique so sensitive? So let's just think about the physics of things for a moment. You have a light source and maybe you have 10 to the 17 photons per second or something like that, right? Coming out of your light source. Let's say you have 1,000 molecules in your sample. And so if you try to detect the absorption of the light, you're trying to detect 10 to the 17th. If every molecule absorbs, then you have 10 to the 17th minus 1,000, which is basically 10 to the 17th. You can't detect the absorption of few molecules. So how is it that we measure the spectrum of samples in an infrared in a mass spectrometer when there are so few ions? We can easily measure 1,000 ions. Well, the answer is that we use a special technique called messenger tagging. So we put a weakly bound tag on the molecule. We typically use nitrogen because it bounds fairly weakly to an ion. And so we look at the mass of a molecule plus this tag. And you can measure all the ions that have that mass. When the molecule absorbs the infrared light, because you've tuned it to a frequency, a vibrational frequency molecule, it absorbs the photon, that energy redistributes, and it pops off this messenger tag that we put on the molecule. And this tag is just something that we attach inside the mass spectrometer at low temperatures. When the tag pops off, the molecule is now at a different mass. It's shifted. In the case of a nitrogen tag, it's shifted by 28 Dalton. And so you can measure the ratio of the tag to the untagged species. And then by measuring that ratio as a function of the laser frequency, we get an infrared spectrum. And so that's how we can do it so sensitively. You're not measuring the absorption of photons, you're measuring a consequence of the absorption of the photons, which we can do with exquisite sensitivity. So I don't know if that was a burning question, but maybe a question that you neglected to ask me, which I think is important.
Wow, that's actually important, and glad you explained it to me, but it's very fascinating how you do that. So as we're wrapping up, Tom, what is the most important takeaway you want our listeners to walk away with?
I think the most important takeaway is that soon you will no longer be restricted to analyzing only a small fraction of molecules in a biological sample. By adding this new data dimension, we'll be able to have a much more global view of all the chemical changes for example, when a drug is metabolized. We'll be able to observe all the metabolites, not just a selected few. And this will change this extra data dimension that allows us to do this will change the way that we do drug development, disease diagnostics.
Fantastic. Tom, where can people connect with you and get a hold of your company and perhaps even explore your services, or once the instrument is ready, get your instrument?
So we have a website. It's www.isospecanalytics.com, or you could send me an email at tom@isospec.ch is the simplest form. And we're also on LinkedIn. Isospec Analytics is on LinkedIn. So through our website, through LinkedIn, there'll be announcements when instruments become available. There was an announcement when we started our collaboration with Agilent, and we hope to announce soon that we're proceeding to the next step.
Well, thank you so much, Tom, for sharing your passion, for sharing your purpose, and also for explaining us how you are changing the analytics space. So it's been a huge pleasure talking to you today and having you on the show today.
Well, it's been my pleasure, David. I'll look forward to listening to more of your podcasts as well.
Thank you for joining me for this two-part conversation with Tom Rizzo. I hope you're walking away with fresh insights on how advanced analytical techniques can solve real problems in your bioprocess development and therapeutic characterization work. If this episode sparked ideas or answered questions you have been wrestling with, I'd love to hear about it. Please leave us a review on Apple Podcasts or your favorite platform. Until next time, keep doing biotech the smart way. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.